So yeah, somehow I guess these previous visits where I always worked with Doug Wallace as well have convinced me to come here permanently, and now I've been here since last year. And I'm going to talk about the first results from the project that I've been working on, which is the export of newly oxygenated Labrador Sea Water following convection. Co-authors are Doug Wallace, obviously, because I'm in his lab, Dasha Atamanchuk, who did a lot of the sensor deployment and calibration and everything for this. Johannes Carstens and at Geoma in Germany, who's supposed to be he is my host supervisor for the fellowship. I was supposed to spend several months in Kiel, which obviously is kind of tricky right now. So far we've just been working remotely together. And then Patricia Handmann, who is one of his postdocs, has been working with us as well. And on the right side, I'm already showing a little teaser from the results, which we'll get back to later. But first, let's start with a little introduction. So even though I'm in a chemical oceanography lab now, I did my PhD in physical oceanography, so I always have to start with that. And kind of to explain why we care about the Labrador Sea. First of all, it's close to Canada, and we all love that. So it's between Labrador and Greenland, and in the center of the basin, there's kind of a convective area. So all these figures are from several of Igor Yashayaev's papers, who works a lot on this kind of stuff. So the line drawn through the figure on the left, I don't know if you can see my mouse, but I hope you can. Actually, it doesn't matter much right now. But this line shows the AR7W hydrography line, which is occupied about once a year. And if you look at the temperature through that section shown in this bottom figure, you can see that there's a very homogeneous layer that occupies the upper 1,000 or 2,000 meters, depending on the year, where the temperature is essentially the same from close to the surface to depth. And the reason for that is that in the Labrador Sea, we have convection happening, which you can also see in the top right figure in this time series of temperature. So each winter, you have the years here at the bottom, each winter around February or March. By the way, I think there's a few people that may not be muted, so don't say anything too loud. But so each winter around February or March, it gets very cold in the Labrador Sea, so the surface water gets very dense, and then eventually gets denser than the water below, and then because of physics, it would like to be below that other water, so it sinks. Then the less dense water comes to the surface, and that gets denser, and then that sinks, and then just keeps going and mixes the water column very quickly to about 1,000 or 2,000 meters, which forms this very homogeneous water mass known as Labrador Sea Water. And then staying with the physics part for now, the reason why we care about that is because the Labrador Sea is one of the few regions in the world where that happens, the other ones being the Nordic seas and near Antarctica, although the process is a bit different. But in the Labrador Sea, we have open ocean convection, and we form Labrador Sea Water, which becomes part of North Atlantic deep water, and that makes up the lower limb of the global overturning circulation, or particularly the Atlantic meridional overturning circulation. So there's warm surface water coming into the Nordic seas and Labrador Sea, and then all that heat loss transforms it to deep water that flows south. And there's been some doubt recently about how important the sinking in the Labrador Sea actually is for setting the strength of this overturning, but there's not really any doubt that it affects the properties. So if you look at the figure on the right, that's kind of a popular example of a salinity section from the Atlantic Ocean, where you can see North Atlantic deep water as a salinity maximum, extending from the North Atlantic, where Labrador Sea Water and some of these other water masses form. And then you can see very nicely that it flows. So at the bottom, you see the latitudes. So it flows all the way to the equator and a bit past that. So now finally we get to move on to oxygen. So if you compare the salinity section to an oxygen section from Schmidtko et al., you see that essentially where you see the salinity maximum associated with North Atlantic deep water, there's also very high oxygen concentrations, particularly if you compare it to the Indian and Pacific Ocean, where the deep ocean has far less oxygen. And then what this Schmidtko et al. Paper really is about is deoxygenation. So they have shown that basically everywhere in the deep ocean in particular, you have the ocean losing oxygen over time. But in fact, the North Atlantic, where we have this North Atlantic deep water, which comes from the Labrador Sea, bringing oxygen to the deep ocean is actually one of the few regions where there's not been any major oxygen loss in the last few decades, which already tells us that that it plays an important role for oxygen. And so the reason for that is that as convection happens and sets physical properties of the water, there's also very strong oxygen uptake. So I always use this overview map on the bottom right from McKinley et al. 2003, which still isn't ideal, but I've struggled to find a better one for showing oxygen uptake. So this very little region near the top that you can almost not see because of the projection is the Labrador Sea. And you can hopefully see that it's very dark blue, which means there's a lot of oxygen going into the ocean in this region. And so because of that, and because of the deep convection, the Labrador Sea is one of the few regions in the world where oxygen from the atmosphere can reach the deep ocean. And this time series at the top is from Dasha's paper, which used the sea cyclone mooring in the center of the basin, showing the oxygen uptake as well as carbon uptake, which you can see is also important. But I'm focusing mostly on oxygen. So in winter, there's this strong oxygen uptake driven by convection together with the fact that there's very strong winds. And then there's a strong air-sea oxygen gradient, and that drives a lot of oxygen into the ocean. So yeah, the Lab Sea is one of the few regions where oxygen can get to the deep ocean, which obviously, since oxygen and carbon are linked together, that's also important for carbon cycling. So my project is part of the OFI module called, I think, Auditing the Northwest Atlantic Carbon Sink, because it's all interrelated, but I'm focusing pretty much only on oxygen. So moving on finally to what we're doing. Well, I guess this is still kind of an intro. So looking at the circulation in the Labrador Sea, this is a rather complicated map, but just focusing on this part on the left, which is where the Labrador Sea is, we have a western boundary current coming in from the Erminger Sea, rounding the basin, then exiting. There's convection happening in the center of the basin. And then this idealized view, there's just Labrador Sea water going in here and then going out. And so there's a mooring array that has been deployed for several decades at this point, I think since the mid-90s, by colleagues in Germany, which is called 53 North. And that mostly has been focused on measuring the current speed. So all of these instruments that you can see in the figure are current meters, and some of them measure temperature salinity as well with microcats. But the colors in this little inset show the current speed through the section, where you can see a deep western boundary current, so strong southward currents at depth, and then the Labrador current over the shelf, and then recirculation in the interior. And so what we have done is to add oxygen sensors to that. So now at the bottom right, I'm showing an oxygen section through there, where you can see a very high oxygen layer in the upper between about 500 and 1,500 meters, which is the Labrador Sea Water flowing out of the basin. And we've deployed oxygen sensors at about 650 meters depth, within that on 4 moorings called K7, K8, K9, and K10. And then there's also sensors deeper in the Northeast Atlantic deep water layer, which I won't really talk about. But so we're using these oxygen sensors at the Labrador Sea Water layer to essentially look at 2 questions, which is how and when the newly formed Labrador Sea Water is exported out of the basin, and then what the oxygen transport associated with that is. So starting off just with time series from all of the moorings, you can see, again, at 650 meters depth, you can see that they all have kind of a similar picture overall, is that starting from May, when they were first deployed in 2016, oxygen slowly decreases over time, then starts to increase somewhere around February or March, stays high for a bit, then decreases again, and then the same thing happens again the next year. So it's about the same for all of the different moorings, with small differences, which I won't get into too much. If you look at the gray lines in the background, so the black is a low pass filtered time series. The black is the high frequency measurements. You can see that it's also a lot of high frequency variability around it, particularly in February and March, which I'll get into in a bit. And then these oxygen changes also occur together with temperature and salinity changes. So looking more at that, oxygen.
Hello. Hi.
Someone is unmuted, I think, just FYI. Oxygen is higher at lower temperature and salinities, but it's not just a solubility thing. So the saturation, or if we redid this figure with saturation, it would be the same figure or the same picture. Same basic idea, I guess. So this figure shows temperature and salinity plots at all 4 mooring sites. So salinity on the x-axis, temperature on the y-axis. And then the colors show the oxygen concentration anomaly, so relative to the mean for each of the moorings. And you can see that they all lie along kind of a diagonal line with almost no cross isopycnal change except for K7. So meaning that the properties change, but the density doesn't actually change that much. We just have either warmer and saltier water or fresher and colder water. And if you look at the 2 kind of prevalent water masses in the region, Erminger water and Labrador Sea Water, almost all of the points lie in between those 2, which, just to give you some orientation, Erminger water is essentially what comes in from the Erminger Sea and is found in the boundary current over here near Greenland. And then Labrador Sea Water is the convective water mass from the center of the basin. So that already tells us that there seems to be some relation, obviously, to the formation of Labrador Sea Water. So then looking at these high frequency changes, so I now zoomed into the oxygen time series, and for this and some of the later analysis, I'll just talk about one of the moorings, K9, because it's easier than to keep talking about 4 different ones that show very similar things. Looking at a 6-month slice of the time series, again, the thick line is the low pass filtered, and then the dots are the high frequency one. You can see that early on, in January and February, there's low oxygen, very little variability around that, kind of stays within a small range. And then all of a sudden in mid-February, you see very large variability, where oxygen concentrations can change by up to 15 micromole in a day. The standard deviation for the 3-month period here is 4 times higher than it is for the 3 months before that. And if you again look at temperature and salinity, so in the top right figure, it's the same idea as the plot on the last slide. Temperature and salinity color coded by oxygen concentration, but now just for 1 mooring and for consecutive 5-day periods. You can see in the beginning of February, you have most of the points up here, high temperature and salinity, low oxygen. Then in the middle of the month, you see these higher oxygen points coming in, and then it just progresses from there, and eventually you have a very large spread in properties from essentially properties that are very much like Labrador Sea Water in the interior to these that are more like what was found in the boundary current before. And so that suggests to us that there may be a lot of or some Labrador Sea Water coming in without mixing too much with the water that was there before, so that it just enters the boundary current and gets advected with it, but doesn't change its properties too much after convection. And then the question that arises from that is, where does that come from? So as I showed or talked about in the beginning in the intro, there's convection mainly happens in the interior, and that's what has been studied traditionally, but there's also been some studies that have shown that it can happen within the boundary current itself. And just to add to that is that we have also looked at whether or not there's any sign at the at our measurement location of convection happening, and there doesn't seem to be. So it does have to come from somewhere upstream, but the question is interior or boundary current. And so to look at that, I've used various products with from Argo floats. So this figure is showing a lot, I guess, but the contours show essentially where convection happens. So it's using the Halty et al. Climatology to look at where or how often at each point mixed layers are exceeding 600 meters, that threshold being used because that's the depth we're at for our measurements, which highlights kind of the classical convective patch in the interior and then also going towards the Erminger Sea. But you can see there's also some onshore of the 3,000 meter isobath. So this thin gray line that shows the just the bottom depth has been used traditionally as kind of the dividing line between interior and then boundary current over the boundary. So you can see that it gets onshore of that a bit, so there does seem to be some convection in the boundary current as well. And then all the symbols in this figure show a different data sets, but also using Argo floats that I basically just downloaded from the Argo database. And I think it's something like 35,000 profiles total that I've looked at. And these symbols are all the ones where convection was measured, which I think are somewhere around 150. And then the different colors or symbols show you what happens with them essentially. So the red dots are profiles where we measure convection, but we don't see any evidence of export afterwards, which we defined as the float being onshore of this 3,000 meter isobath and then staying there for about a month. The pink ones are the ones that are exported, and then the green ones are the ones that are exported in either January or February. So first of all, that shows that a lot of water that's convected in the interior doesn't actually leave the region in the same year, which is something that other studies have shown as well, that there's a significant fraction that just stays in the center of the basin. Whereas if convection does happen in the boundary current, even though it's less likely or less common, then the water tends to be exported as well because it's already in the boundary current, so it doesn't need to go anywhere essentially. And then on top of that, the green symbols, as I said, show the ones where export occurs early on in the season. And as I showed in the last slide, we see an increase down here, which is where the moorings are, is this black line, an increase in oxygen in February, which suggests that the Labrador Sea Water arrives there then. And really the only ones that are anywhere close to that are these two floats over here. So the ones that are either extremely close to the boundary or within the boundary current. which suggests that this initial increase in oxygen that we see at least comes from convection within the boundary current or close to it. Okay, so then moving on to the figure that I started with, we now want to look at the seasonal cycle of oxygen and of properties, which I suppose there's a lot going on here in this figure because it's 3D. So we have oxygen on the x-axis, then time on the y-axis going from December to the next December, and then just the number of observations and the z-axis, and then we have colors as well. So it's essentially just a histogram for each month of how many observations are within each oxygen bin. And then the colors show a water mass index that are defined based on, I can just jump back to this slide, based on essentially where along this mixing line between Labrador Sea Water and Erminger water the measurement lies. And so it just shows you that for about half of the year, we have something that looks more like this Erminger water that is found near Greenland. And then for the other half, we have something that looks more like Labrador Sea Water. And as much as I like this 3D figure, it's a bit easier to look at it in kind of a top-down view. So it shows the exact same thing. But now instead of having the peaks, the size of the symbol is how many measurements are within each bin. And then the black line shows you the maximum or the most common oxygen concentration measured for each month. So just to walk through this annual cycle, we start off in the beginning of the year or the end of the first year, beginning of the next, with low oxygen, high temperature and salinity, and at least for these 2 months, very low spread around the mean concentration, so relatively homogeneous water. Then, whoa, I just went back accidentally. Then around mid-February, as I showed in the last few slides, we have this arrival of Labrador Sea Water. So you see some bins here emerging with some more measurements in them, and a very large spread between the highest and lowest measurements for the months. And then in March, it becomes Labrador Sea Water becomes the more common water mass. Then around April to June, we have mostly Labrador Sea Water being affected through the section. Then July to September, we have transitioned back and have, again, both of the water masses present in varying quantities. And then October to December, we kind of go back to where we started with lower oxygen values, less spread, and temperature and salinity that are more like those of Erminger water. And so we can also compare that to the fraction of convected Labrador seawater that's exported. So that's based on this map that I showed a few slides ago, where I looked at the where convection happens and when convection or when these floats are exported. And if you just compare the shape of that to the shape of the oxygen time series, they look very similar, but with a delay of about a month. So sorry, I went backwards again. There's a delay of about a month between the two time series. So we see the amount of water entering, of Labrador Sea Water entering the boundary current increase around mid-January to early February. A month later, we see the oxygen increase. Then the peak is around mid-March. About a month later, there's the peak in oxygen, and then it starts to decrease again in June. And again, a month later, we see the same kind of variability. So this 1 month delay is consistent with how long it will take approximately from where the water enters the boundary current to 50 north, or at least from the closest location to 50 north, which suggests that the properties of the water leaving the basin are controlled by how much Labrador Sea Water enters the boundary current, at least at this step. And then one more thing to compare to, again, using another one of Igor Yeshayaev's figures, I had to mirror this several times to make it comparable, so the text doesn't make any sense on it, but just to compare again the time series. So this is essentially the same, measures the same thing as our export fraction on the left from floats, but from a different method, using the layer thickness in the interior of the basin to see how much Labrador Sea Water is exported, and it just shows that it matches it pretty well between these two time series. So then the implication of that, I had tried at some point to make a more sophisticated schematic, but failed miserably. Apparently it's pretty difficult to make schematics. But for now, I'm just doing a simplified one, is that during summer, you have this direct export of Labrador Sea Water that just been convected into the boundary current. And during winter, you don't. To be a bit more accurate, you should probably, there would also be probably more Erminger water. So it's the green arrows from the boundary entering the interior. And in fact, it's worth noting, or it should be noted, that the overall transport through the section, I haven't looked at that myself too much yet, but based on other studies that have used the data, there's not really any seasonality in how much water overall is transported out of the basin. It's just different fractions of these different water masses. And so, again, Labrador Sea Water, or water with properties like Labrador Sea Water, mostly leaves during the months following convection, and then it looks more like Erminger water during autumn and winter, at least at the depth that we're measuring at. And then based on the data, I'm just going to jump back a slide again. So we also did a rough estimate of the oxygen transport just due to this change from these lower oxygen values to the higher ones. So due to the input of Labrador Sea Water. So essentially just integrating the area under this curve and multiplying by mean transport. And that would result in an export of about 1 teramole of oxygen, which is about 45% of what's taken up in the interior. And so you may now ask yourself what happens to the other 55%. And I'm glad you asked. That's part of what we're hoping to get at in the future. So looking at this kind of schematic spreading pathways, again, from one of Igor Yeshayaev's papers. Currently, I really like using his figures. We are essentially only looking at the southward export of Labrador Sea Water, but there's also some going into the Erminger Sea. And then I added this green arrow to show that that eventually comes back around, actually becomes part of this Erminger water that we used as our baseline. And then convection also happens in the Erminger Sea itself. I'm shown here in this potential vorticity map from Femke de Jong. So once we account for that and these different pathways, the real export may be several times higher than what we've shown here. And so one of the ideas or plans for the future is to combine the oxygen data that we have with the velocity data and get the full time series of the varying oxygen transports through the section. And then to also use CTD data from the GeoVeed cruises to try and get a net transport, because really what matters is how much more oxygen leaves the Labrador Sea than comes into it with the surface flow. And then one thing that will also help with getting kind of the bigger picture is the fact that there's now a lot more oxygen measurements going on in the region than there was at the time that I'm using the data from. So we've added more sensors to the 53 North moorings, and then this top figure shows the 53 North array is actually part of this much larger project called OSNAP, which until recently was pretty much purely physical oceanography, but now some people, in particular Jamie Poulter at URI and Isabelle Lebras at HUI, have added some oxygen sensors to other parts of that array as well. So on the western side of Greenland, in the northeastern Labrador Sea, and then on the eastern side of Greenland in the Erminger Sea. And so all these, I think all the red and pink dots in these figures show oxygen measurements. But we now have 3 different sections with 70 sensors total, and everything I've shown so far is based on 4 of those. So I think once all of that is recovered, we will have a much better idea, and we'll have much more data to look at some of the more complicated questions about these pathways. Okay, to summarize, we have measured oxygen at the exit of the Labrador Sea through the section at 53 North. We have shown that oxygen levels increase due to input of Labrador Sea Water, which is formed in the interior of the basin by convection, but also close to the boundary current, which is responsible for the initial increase. And then most of the direct export of newly ventilated Labrador Sea Water happens from late February to August. And then about 45% of the oxygen taken up in the interior in the same year is exported through this region during that time. And that is all. So thank you for listening, and we'll take any questions you have.
Thanks a lot, Yannis. Are there any questions out there? Just jump in if you have one. Jinyu.
Very good. I really enjoy your talk, and I'm a physical oceanographer. Clearly, do you hear me? I really like your talk, and so my question is clearly the observation, either is Argus or buoy data are really very highly valuable. Okay, that's no doubt. And my real question is look at the high frequency variability and also kind of a long trend of oxygen problem in Labrador Sea. I wonder how is it important to have a model. I know you told me the observation analysis. I'm quite happy with that. I just wonder what can you do if you have a model to answer some specific question you have, such as the advection and the convection to influence oxygen, and particularly maybe the biological process may play the role. I don't know how much is the percentage. Yeah, so that's my question.
Yeah, it's definitely a very good question. And I mean, so the Labrador Sea is, one of the regions with the best coverage from Argo floats, but even then, I think it's still not enough to get, a whole spatially varying picture and seasonal variability and all that. And then on top of that, I'm gonna go back. So this slide that I'm showing with the export of Argo floats, I'm doing kind of a simple analysis based on where floats go after convection, but there's people that have done this a lot more thoroughly using model data. So it's things like the pathways of Labrador Sea Water can be studied in more detail with models, and then all these biological processes and oxygen. There's definitely a lot of things that if we work together well with observations and models, then that can be very useful to answer all these questions.
Okay, all right, thank you, good.
Yo-Yu, I think you have a question.
Yeah, a question for the last point for your summary, your conclusion. And from the tracer point of view, how do you decide what is exported, what is remain inside in the domain? So you said 45% is exported. How do you decide that without velocity or measure, just from tracer point of view?
So is your question where the 45% comes from, like how I decide how much is taken up?
Yeah.
Oh, that's just so based on the sea cycle data from the interior, so we go all the way back to this figure from the Atamanchuk et al. Paper. So it's basically this uptake that we measured in a different study, multiplied by the area of the Labrador Sea, gives us an estimate of the uptake in the interior. And then the transport, the oxygen transport out of the basin is about 45% of that number.
Okay, you make use of velocity. You must use the velocity information, right? Use a flux.
Yeah. So I'm using velocity, but I'm not using time varying velocity, basically.
Okay.
So the plan is, or one of the plans for this project is to eventually get, a full gridded oxygen field and a full gridded velocity field and just multiply them to get transports, but I'm not at that point yet. So for this, I just used the mean velocity and section width and the increase in oxygen, and then that gives me an oxygen transport. Which is why I think in the slide I said it's a rough estimate.
Sure. Okay.
So it's just the first number to kind of get an idea.
If it's okay, David, I've just got a quick
Yeah,
question.
go for it. Go for it.
Have you had a chance, Yanis, yet to look to see if there's any hint of covariance of the velocities and the oxygen concentrations during that winter period, or which, or
Well, I don't have the figure ready right now, but I had looked at, so one of the moorings, it does seem like basically during or right after convection, as new Labrador Sea Water is coming in, the velocity is more barotropic, so almost constant in depth, and then during the rest of the year it's more baroclinic, which I think was the K8 mooring that's close to the edge of the Labrador Current and the deep western boundary current, but I haven't calculated any covariances yet. But part of that is also, it gets tricky because obviously there can be current meanders and things like that. So just because velocity varies doesn't mean the real throughput varies. It might just be that the current moved to a different part, and we're not measuring the same section of it.
Any other questions out there for Yanis?
Hey, Giannis. This is Jamie. That was great. I'm back at one question to understand that export rate, how you do that again. So you take the time average velocity, multiply the width of the section, and then you multiply by the delta oxygen of like the max in winter minus the minimum, sorry, the max in spring minus the minimum in winter. Is that right?
Yeah.
Okay. So anything, if there's any like leakage during the summer, that makes it that's not in there. It's the export particularly due to the gain of oxygen in the winter season. Is that right?
Yeah. So it's basically, I think I called it the direct export of oxygen. So it's, again, going back to this figure, it's basically what's added just by Labrador Sea Water from the center going south. Whereas the kind of the baseline that I used for the difference in oxygen, that will already be elevated as well, because we have Labrador Sea Water going into the Erminger Sea and coming back, and we have convection in the Erminger Sea. We also have Labrador Sea Water entering the boundary current to the north. So all of that will affect the oxygen concentration too. So it's basically a very simplified first estimate of just this one pathway.
Yeah. It's still great progress. It's so cool to see the data in action.
Yeah.
Thanks.
Will be a lot more to come once all the other measurements come back.
okay, I've got another one, David. Am I allowed to ask a second question?
Absolutely.
Because I agree with Jamie. It's really great to see the data and just makes your brain race and thinking about it. So I think the one thing we were surprised at was, and maybe we shouldn't have been, but we are surprised at that high variance, you know, and that lack of mixing in the boundary current is what I'd like to say. Is it going to be possible to get an idea, and I'm sure you haven't had a chance to do it yet, but is it going to be possible to get an idea of the scale of those high oxygen patches, the length scale of those high oxygen patches, or do you have any feeling for, is that kind of centimeter scale, kilometer scale, eddy scale? Do you have any feeling for that?
I mean, yeah, it's going to be difficult. I haven't looked at it yet. I think it's going to be kilometer scale, but not a lot of kilometers. But then again, part of what's difficult with that is, so I think I'm going to go back to this slide for a second. So all these kind of wiggles around the mean increase don't seem to be correlated between moorings. So it's not like one patch comes through all of them at the same time, but that sometimes it appears at one of them, sometimes at another. So again, it can be that that a patch seems to disappear, but it could have just moved away from where we're measuring. So that will be tricky to get a good estimate from, but yeah, I'll see what comes out of that. Well, my guess would be tens of kilometers scale, maybe.
Okay. Well, thanks a lot. That was very interesting, Hannes.
Yeah. Thanks for having me.
Thanks everyone for coming.
Thanks so much. Bye.